Okay, the recording has started. We're going to start. Okay, this is the first question. As you can see, it's from November 2024, so it's quite recent. The diagram below represents a DNA molecule undergoing an important biological process. So the people that are going to be assisting us with answering this, Jonita, Kolile, Wavudi, the three of you, please work together in answering this question. Identify the process shown in the diagram above. That is 141A. Jonita?
Okay, ma'am. Is DNA replication?
Thank you so much. DNA replication. So I might not be able to write the full words, guys, because my device becomes very slow once I use a lot of things on Team. The mic, the camera, the recording, projecting, the slides or the PDF. So, but please do listen to the answer. That is DNA replication. Thank you so much. Then the chemical bond labeled 1. The next person that we're working with, there's 3 people there. So Polile, then Wavudi, you are next.
Hydrogen bond.
Hydrogen bond. Thank you so much. So that will be the hydrogen bonds. It's not going to write properly. Hydrogen bonds. Okay. Then give the collective name for the parts labeled 2, 3, and 4. Wavudi?
Nucleotide.
Nucleotide. Thank you so much. This would be the nucleotide. It's not going to come to the party. Once I start recording, it does this. Okay, so full name for the nitrogen base labeled 5. I think let's just listen more, then you can make a note on your side. Nitrogen base labeled 5. There it is. So who's answering for us next? We can still continue with you guys, Jonita, Kulile, and Wavudi. Anyone,
It's
please?
cytosine.
Cytosine.
Thymine.
Thymine. Which one are we taking, guys? So we've got 2 options. There was one person that said C, and another person is saying T. So which one is the correct answer? The other people, you can also unmute if you want to add. So there's 2 options on the table for us. So we've got C and T. What will be the correct one?
Thymine, ma'am.
Thymine is correct. Thank you so much. It will be thymine. So the full name of the nitrogenous base labeled 5 is thymine. And remember, for this one, you have to write the full name because that's what they've asked. So I'm not sure how we confuse this with C, because if you are looking at the strands here, this would be your template strand and this would be your template strand. Now, if you are looking at these bases from the bottom, there is a G and a G is paired with this base. And then there is a and A will be paired with this one, which will be T, which is thymine. So I think maybe there was just a mistake here in looking at the basis. Okay. Then during which phase of the cycle does this process take place? Yes, I'm listening.
During interphase.
During interphase. Thank you so much. So 1.4.3 is during interphase. Where in the cell does this process occur? So where exactly in the cell will it occur? Olile?
In the nucleus.
In the nucleus.
Nucleus.
6
Yes.
nucleus. 6 marks. 6 marks. I think all of us got 6 marks here. I think if there was a mistake, there was just that mistake at 142B. But I think this was clear. So think of it. If you're writing a November 2024 paper, this could have been your question. There was no way that you're going to fail this because it's straightforward. Okay, we're moving to the next question. I like this one. We are going to work a little bit. We are working for 12 marks here. Okay, so we are going to have more people helping us. I'm seeing Tsepiso, Jakida, and Lelam. So the three of you, let's please work together. But this is protein synthesis. So I'm going to be asking more people to come on board to also assist with this one. All right, let's look at it. The diagram below represents a certain stage of protein synthesis. Now, they also tell you that we are reading the bases from left to right. That is very important for us to note. Okay, if I'm still reading a question, I will need the mics to be muted. Then once I start asking for answers, then you can unmute. Okay, thank you so much. So my point was the sequences in this case are read from left to right. I would say 99.99% of the time, you should be reading them from left to right, unless otherwise stated in an exam that you are reading them from right to left. That would be confusing, but hey, you just need to follow what you are given. Now, you need to be able to analyze this diagram before you even go to your questions. So what we are having here, they're saying, okay, this particular part or this diagram, actually not even the part, this structure over here is molecule X. They're not giving us the name of the molecule, but then they show us the section of molecule X whereby we are able to see bases, or these lines represent bases here and bases there on this strand. Now they give us the names of the strands. They say this is strand 1 and then this is strand 2. Between strand 1 and strand 2, they tell us that strand 1 is a template. That should ring a bell for us. Okay? And then between these two strands, there's a strand or a molecule that has now been formed, and they're saying that is molecule Y. So between the two strands, they've used bases of the template strand to form molecule Y. Me and you should know exactly what is happening here. 211. Identify molecule Y. Tepi? Molecule Y, Tepiso?
It's an molecule Y is an mRNA.
Definitely an mRNA. I'm trying my best to write, but if it's not coming to the party, I'm just going to leave it. Okay, so it's mRNA. That is clear. It should really, really, really be clear. If you are confused about anything, please unmute anytime and ask so that I can clarify. Then 212 for 6 marks. Ooh, I like this question. Describe the process that resulted in the formation of molecule Y. Describe the process that resulted in the formation of messenger RNA. For how many marks? 6 marks. We're not skipping this in an exam because this is a copy and paste from our notes. Jakida, please do help us out with the 6 marks.
Okay. My answer is the DNA strand, the DNA double helix unwinds, the two double strands unzip, hydrogen bonds break to form two separate strands. One strand is used as a template to form mRNA. Free RNA nucleotides in the nucleoplast pair with the like the bonds, yeah, to form the mRNA. The mRNA is
Complementary.
complementary to the DNA.
Okay, let's start from the first line. Give me your first line.
The DNA double helix unwinds.
1 mark. DNA double helix unwinds. 1 mark. The second one?
The DNA strands unzip,
Okay.
hydrogen bonds break.
Okay, so you tell me what you think will come first. Do you think the molecule will unzip first and then the bonds break? Or do you think the bonds will break, resulting in the molecule unzipping?
The bonds will break, resulting in the molecule
you.
unzipping.
Thank you. So that means in the second point, you were going to lose a mark because you said that the molecule unzips and then the bonds will break, and that is incorrect. You would have swapped it around. So please make sure, guys, that the order of the points is correct for you to get a mark. So it's that DNA double helix molecule unwinds, and then the bonds in between the strands are going to break, leading to the molecule unzipping. The molecule is only unzipping because the bonds are breaking. And then I think the rest of your points were correct then. So forming two separate strands, one strand is going to be used as a template to form a messenger RNA strand. The messenger RNA will be complementary, the messenger RNA will be complementary to the template strand. I think by the first 5 points, you would have already gotten your 6 marks. So that is good. I think that's all I had to clarify. Also, something to note here, the question is asking you to describe the process. So if you decide to mention the name of the process, you're not going to get a mark. So you can mention it if you want, but I normally say mention it in your brain to say, okay, they are asking me to describe transcription. So that's the process that I'm describing. So another thing that is important, using using RNA nucleotides from the nucleoplasm. So if you said DNA nucleotides, you were going to lose a mark for that. But I think everything that you mentioned was correct. I just had to fix that. Anyone who wants to ask about their 2.1.2 process? Anyone who wants to ask? Okay, I'll move. All right, then remember, guys, if you are raising a hand or anything, I can't see you because I'm looking at my question paper now. So just unmute any second and then ask. So 2.1.3, give one structural difference between molecule X and molecule Y. Give one, and they're telling you the difference that you should be focusing on. They said structural in terms of structure. Molecule X, there it is, and molecule Y, there it is. Let's see who's answering for us. Lelam Mayekiso. Yes, I'm listening. I can't hear you, Lelam. I can see your mic is unmuted, so I'm assuming you are speaking, but I can't hear you. Okay. Sandiswa, you are next on my list. Sandiswa, please do answer that for us.
Anti-codon pairs with codon of a messenger RNA.
Okay, I didn't hear the first part. What did you say?
Anticodons, ma'am, pairs with a codon of a messenger RNA.
Okay, let's look at this. Let's break down this question so that me and you can understand it. So in 2.1.3, they're saying we must give one structural difference in terms of the structure. And the difference that we are looking at is between molecule X and molecule Y. There is molecule X here. Let me try and change my color. There is molecule X here. This is what they've labeled. There's molecule X actually with the two strands. There is molecule X. And molecule Y is the strand here that is formed in the middle. So let me first ask you this before we get to the question. What do you think molecule X is? Sandiswa?
I think,
yes.
ma'am, it's the transcription of the RNA.
Okay, so when you see the word molecule, you must think of only two things, DNA, RNA. And then remember in RNA, we then have the messenger RNA and the transfer RNA. So when you see molecule X, do you think it's DNA or RNA? When you see molecule Y, do you think it's DNA or RNA? So let's start here with molecule X. Do you think it's DNA or RNA? Just look at the shape here.
DNA.
I think, ma'am, it's DNA.
Thank you so much. It's DNA. Remember, we have not answered the question. I am now trying to lead you to answering the question. Then molecule Y. Do you think this molecule, this is the molecule here that's formed in the middle, the one that I'm highlighting in red. Do you think that one is a DNA or an RNA? We've actually answered it here. We said this one is an RNA, and to be specific, it is a messenger RNA. So the question is asking you the structural difference between DNA and messenger RNA. So now, the most common answer, or the most obvious one, you can speak about the strands. With molecule X, you have two strands. There is strand 1 and there is strand 2. So molecule X is made up of two strands, or has two strands, and molecule Y has one strand. That's the structural difference. Anyone else with an addition there or a different answer?
Can I help, ma'am?
Yes.
Okay. So eyokuqala ngithe in mRNA it has only single strand. Le enye ine double strand, DNA. Then
Yes.
DNA ine lokhunjana ine deoxyribosome sugar and le ine ribosome sugar. And in RNA it has a nitrogenous based uracil le enye ine thymine.
Okay. Let me
Hi,
just
ma'am.
see who was answering. I think it was
Nonhlanhla.
Tepiso, right?
Oh, okay.
Hi, ma'am.
Yes.
Can I just ask a question regarding the question? It says, give one structural difference. So when it comes to structure, is it not only about the shape of the actual molecule?
What do you mean shape? What do you mean by shape?
Like physical appearance, I guess, like maybe that it's a double stranded helix.
It's a double-stranded, yes. So you need to be able to have an answer where you can thoroughly compare. So if you say molecule X is double-stranded and molecule Y is single-stranded, that is correct, because you're speaking in terms of the structure. Is that fine?
Yes.
Okay, I want to go back to Tepiso. I think it was Tepiso that was answering. Tepi, you mentioned something about the sugar, right? Yes, is it you, Tepi?
It was Nonhlanhla.
Nonhlanhla. Oh, okay. Thank you
Yes.
so much. Sorry. Nonhlanhla, you mentioned something about the sugar. This is when life science becomes tricky and they try and confuse you in order to penalize you. Me and you know that between DNA and RNA, DNA will have a deoxyribose sugar and RNA will have a ribose sugar. But will that be a structural difference? No, it won't be. Is it a difference between DNA and RNA? Yes. But here they were specific what difference they wanted. They wanted a structural one. So you are comparing the structure. One is double-stranded and the other is single-stranded. So the ribose sugar and deoxyribose sugar would be marked incorrect when answering this specific question. If they had said, let's tabulate differences, without directing us what exactly we should differentiate, then we can accept the sugar business. I hope that makes sense, Nonhlanhla?
Yes, it is.
Yes, thank you so much. And I hope that makes sense for everyone. Another difference in terms of structure, this one can be tricky because people can argue that it is part of structure, or other people can argue that it's not part of structure. The thymine for molecule X and the uracil for molecule Y. Oh, it's not in the memo. I think the argument would be if we're asking about the difference in terms of bases, then you would mention that. Here they were speaking about structure only. I think we did add something to say, okay, molecule Y is normally shorter in terms of structure, and then molecule X is normally longer. I think they were accepting that. One thing I need to mention, in your answer, you need to tell us about molecule X and molecule Y. If you just say one is double-stranded and the other is single-stranded, you lose the 2 marks. Because what are you talking about? What is one and the other? We don't know what that is. So whatever that you are asked in a question, make sure you reference it in your answer. So you have to say molecule X is double-stranded so that we know, oh, okay, you are speaking about X in the double-stranded point. But if you say one is double-stranded, the other is, or it is double-stranded, no mark for it. Okay, I hope that is clear. Then give the sequence of bases at triplet Z. This one I'm interested to know. This one, I will say it's an open line. I'm going to take 4 options. This one, I am very, very, very much interested to know. Give the sequence of bases at triplet Z. There is triplet Z, and it's in a DNA strand. On strand 2, they are telling you on strand 2. I am taking 4 options. If we do get to 4, let me say 3 options. Please, anyone unmute and tell me your option. No, this pen. Anyone can unmute and tell me their option.
A, U, G.
So the first one, let me see who answered. I can't see who answered. Please answer again so that I can see on my screen.
A, U, G.
Okay. There's 4 dots. There's no name there.
Oh, sorry, I was late when I was writing, so I didn't just read dots because I was late.
Oh, okay. So you're saying it's A. So please be patient with me, guys, because I have to write very slow so that the pen can pick that up. A, U, G. All right. So this is the first option that we have, guys. A, U, G. So that's a G, but like I'm saying, my pen is going to act up. Please. Okay, let me get the second option for an answer. Give me yours, anyone. I can see Tandazo, you ready to speak?
Yes, ma'am. It's TAC.
TAC,
Yes.
TAC. Okay, this was a G, guys. I'm trying to fix it. Okay, then I'm ready to take the third option. Anyone else with different bases? There should be different bases. I can see Nontlantla, or is it Junita who's trying to speak?
Yes, ma'am. It's A-T-G.
A-T-G.
Okay, I was going to say the same thing then.
Oh, okay. Okay, ATG. Okay, there's a few that also got ATG. Anyone else with a different triplet? Please just unmute and tell me your triplet. We should have another option. Anyone? Okay. Let's look at it, guys. What do we have here? So we are having transcription taking place. So you have strand 1, which is the template strand that was used in order to form these bases for messenger RNA. Strand 2 is our non-template strand. So the bases there were not used. Right. So in order for us to find the triplet for Z, we actually need to first find the bases for strand 1. Why is that? Because these bases here, these 3 bases over here, are the bases that were used in order to find UAC. So we cannot just use UAC and match UAC with the bases here. That will be incorrect because they didn't use strand 2 at all. This was a guideline. In order for us to form molecule 1, which is the messenger RNA, they used bases on these strand. So now let's find these 3 bases first. If we're having a UAC, why are we looking in line with the 3 on strand 2? So UAC, you are going to have A here. So that will be A. And then that will be T. And then that would be G. Right? So these are, that's not our answer. These are the bases of the template strand, A, T, G. Now that means in order for this messenger RNA strand and these three codons to be formed, they use the code from these ATG. Now let's remember, if this is our template strand and this is our non-template strand, the bases that we have here were initially paired with the bases here. So we are going to use this ATG to find bases at Z. So A was initially paired with T. I hope it makes sense. And then T over here was initially paired with A. We are just using the base pairs. And then G over here was initially paired with C. So which one is our answer? B, TAC. Now you could not just look at UAC in the messenger RNA strand and find the Z triplets. That is incorrect. And for option A, there's a U. That makes it obvious that it's incorrect because now this triplet here is in a DNA strand. We cannot have a U in a DNA strand. So this was a bit tricky because you first had to find bases for the template strand and then use those bases in order to find the ones for strand 2. I'm happy to take questions for this one because I knew that some people will be confused by it. That's their goal, to try and confuse you. I'm happy to take questions. You can unmute and ask a question if it's not clear. So I'm not sure how we got ATG. Oh, okay. I think ATG would be almost the same as this one. So the person would have just used the bases here, UAC, and then tried to find bases for the strand. Okay, that would be incorrect. Is it clear, guys? I need a yes or a thumbs up or something for us to move. A yes, a thumbs up, something, something. All right, okay. So please don't feel bad if you are getting these answers wrong. I'm happy for you to get them wrong in the lesson. But if you get them wrong in a test, that's when I won't be too happy. So you can get them wrong here. Then you know what, when you do the activities again, you'll get them right. I'm getting a comment that I must explain again. Okay, so if we're to take it back to transcription, how we cover transcription, remember we said that you will have 2 strands of DNA. You will have this DNA strand and another DNA strand. These are the 2 strands of DNA. Between those 2 strands, we are going to use only 1 strand to form bases, right? For the messenger RNA strand. Now my pen is going to be too slow for me to have a proper example, but if these are the bases in our 2 strands, for example, okay, so if this one is our template strand, I'm putting a dot there to just show that this is the template strand. But these 2 strands were once bonded together by weak hydrogen bonds. So if my bases, for example, is TGC, that means the bases on the other strand would obviously be ACG. But we said we are using this particular strand to form the messenger RNA. I'm going to try and change my color here to form the messenger RNA. Now the messenger RNA will be here in the middle, and we are going to use bases on the left strand in our case, because I've said that's the template strand. So now T will be paired with A, since we are forming a messenger RNA strand. G will be paired with C, and C will be paired with G. This strand will be the messenger RNA strand. So what do we have here in our example? In our example, they are asking you for this, which is that Z, on the non-template strand. But remember, we used the template strand bases in order for us to find the messenger RNA. So we're not going to use the messenger RNA bases to find the non-template strand. We're going to use this one to find the bases the other side. So that's how it works. So this was a bit tricky. That's why I said at the beginning, I want to hear people's answers. But yeah, that's how you work it out. So what I've drawn here and what is in here is actually the same thing. So the Z is this side, the bases. Then the ATG is this side over here. Then this UAC is this middle strand with the bases. Okay, that's protein synthesis for you. I'm burning. Okay. The table below shows some amino acids coded for by codons of molecule Y. We need to then remind ourselves what a codon is. Now, a codon is 3 bases in a messenger RNA strand. Me and you have confirmed that molecule Y is the messenger RNA. So now we are going to use the codons here and read them from left to right in order for us to then find the amino acids. So what are we going to do? We are going to say, oh, okay, there is AGA, that's the first codon. And then AGA, and in our amino acid table, they are also using codon. So we don't have to work anything out here. We are going to say, there is AGA. Let's look for AGA in the table. There is codon AGA. What's the name of the amino acid? Arginine. You write it down. Because what did they say? I didn't read the question. This teacher, oh my God. Identify the first and last amino acid. That's all they asked for. Okay, I guess I wasn't that wrong. The first amino acid will be determined by AGA. Remember, we are reading the codons from left to right. So this would be the first codon. UAC would be the second codon. CCA would be the last codon. In the question, they are specifically asking for the first and the last. So you then indicate in your answer, this is very important. You then indicate and say, first, you write it down. The first one is arginine. Write the word first, and then you give us the amino acid, arginine. Where am I getting this arginine? There it is. I'm not getting it anywhere else but the table because it's the one that is coding for AGA. Then that means we are done with the first amino acid. Let's look for the last amino acid. You go back to your molecule Y, CCA. You then look for CCA on the table. There is CCA. What's the name of the amino acid? Proline. You then write last.
Proline.
Thank you. There you go, guys. My pen is not coming to the party, but yeah. Anyone with a question for this one, guys? This is November 2024. Anyone with a question about anything on this? All right, we move. Okay. Let's get someone to help us with this one quickly. Quickly, this is not protein synthesis, so this should be quick. Anyone? We have people that have not spoken. Let's see, let me get more names here. Okay, Grinam, Lwazi, Lulu, Boikanyo, Dalo, Nomdaniso. I haven't heard your voices, guys, so let's work on this one. The diagrams below represent 2 types of nucleic acids. 2 types of nucleic acids. Identify the nucleic acid shown in diagram 1. There's diagram 1. What is the type of nucleic acid there? Anyone?
DNA.
DNA. Okay, thank you, guys. There's Lulu, there's Dalobutle. Thank you so much. Diagram 1 is definitely DNA. Then identify A and B. There is A over here, and there is B over there. So let's see. Dalobutle, what did you say for A?
A is weak hydrogen bonds.
Weak hydrogen bonds. Remember, if you didn't write the word weak, that is still correct. Hydrogen bonds. Lulu, what did you write for B?
For B, it's phosphate.
Phosphate. That is enough for you to get 1 mark. So whether you wrote the word group or not, it doesn't matter. Phosphate is enough. Okay, name the type of sugar found in the nucleic acid represented in diagram 2. The type of sugar, Gina, please. Gina, the type of sugar found in diagram 2. Okay, Nomtaniso, I'm listening.
Ribose sugar.
Ribose sugar. Thank you so much. That is ribose sugar. So we've answered this. I'm not writing down, guys, because my pen is slow and I want us to cover as many questions as possible. So ribose sugar. Then which diagram, 1 or 2, represents a nucleic acid that is used for tracing of human ancestry and paternity testing? Which diagram are we using? Diagram 1 or 2? Boikanyo?
You use diagram 2 for tracing of human ancestry.
Tracing of human ancestry, you're saying we're using diagram 2?
Yes.
So when we're looking at an ancestral lineage, do we look at people's DNA or people's RNA? You tell me.
Is it their DNA?
It's their DNA. So between diagram 1 and 2, which one is representing DNA?
1.
Thank you so much. That's how you look at it, guys. Diagram 1. Then paternity testing. When we want to identify the father, do we look at his DNA or do we look at his RNA? Lwazi?
DNA, ma'am.
DNA. So which diagram is DNA?
Diagram 1.
Thank you so much. 6 marks. This was from June 2024, 2 years back. 6 marks that we can easily get, guys. Okay, I also like this one. We are back to protein synthesis. I love protein synthesis. Okay. So anyone can unmute on this one, so that at least we just move a bit quicker. So let's see. The diagram below represents a stage during the synthesis of a certain protein, during the making of a certain protein. Now, what are we able to see here? You should be able to analyze your diagram before you even look at the questions. We are seeing something here. This represents a particular organelle. Then on this organelle, there's this strand that is attached here. I wonder what the strand is. And then we are having another molecule here, and then there are bases. We have some of the bases, and then some of the bases are not there. And here we've got names. I wonder what these names represent. Obviously, we can see what is happening here. Me and you should be able to tell what is happening here. Anyone, identify organelle 3. This is organelle 3. What is organelle 3?
I think, ma'am, it's ribosome.
Ribosome.
Thank you so much.
Ribosome.
Thank you. Ribosome. That's definitely the ribosome. 1 mark. For 2 marks, describe the role of molecule 1 during this stage of protein synthesis. We're not going to skip this. We don't skip any question. Even if you feel like the question is difficult, you need to try your best. Now, the question is asking us to describe the role of this molecule. But before we describe the role, me and you, in our minds, need to determine what that molecule is first. And that molecule is transfer RNA. Look at that shape. So they are asking you the role of transfer RNA in that particular process. Okay, the molecule is transfer RNA. Which process is that? And that is translation. So they are asking you the role of transfer RNA during translation. Wavudi, what did you say?
I haven't answered that question yet.
Okay. Anyone who has answered, guys? I'm gonna wait for an answer from your side. If there's no answer, we are going to wait here. If there's no answer, we'll wait. Anyone can unmute and give me the answer.
It carries a specific amino acid to the ribosome.
2 marks. Full 2 marks. It carries a specific amino acid. If you don't have the word specific, wrong, because it doesn't just carry any amino acid. Now, Yes, as much as I'm giving you 2 marks, but I want you to add a little bit of sauce in order for you to get full 2 marks. It carries a specific amino acid that is determined by the messenger RNA codons to the ribosomes. Because remember, in order for it to know which amino acid it needs to pick up in the cytoplasm, it is going to be guided by the messenger RNA codon to say, okay, if the codon is UAC, that means my anticodon will then be A. I don't remember my bases that I mentioned, and then this will be the amino acid for that. So it carries the specific amino acids coded for by the codons of messenger RNA to the ribosome. Full 2 marks. Thank you for that. I'm not able to write it down, but please, if you did listen, please do write it down. Carries the specific amino acids to the ribosome, but obviously you need to include coded for by the messenger RNA codons to the ribosome. That's how you get your 2 marks. Give the sequence of nitrogenous bases at 2. Where is 2? There is 2 over here. For 1 mark, give us the sequence of nitrogenous bases. I don't want people to join the meeting and not talk, guys. Okay, CCU, Lelam has answered CCU. Let's figure it out. So the bases at 2 are basically anticodons. So now those anticodons will be complementary to the codons here in the messenger RNA, because me and you should already know that this strand over here is the messenger RNA. So these would be the anticodons for the GGA here. So it will definitely be CCU. That is correct. So the sequence of nitrogenous bases at 2, definitely CCU. I hope you got that 1 mark. Like I said, stop me any second and ask a question if there's something that is unclear. Then also give us the sequence of nitrogenous bases on the DNA molecule that coded for histidine. They want you to work, because look at the mark allocation here. It's 2 marks. Look at the mark allocation for 213A. It was 1 mark. So this one was easy. We are just looking at the messenger RNA codons. We get our anticodons, we write them. So for 2 marks, that means we got to work a little bit. So where are they directing us? They want a DNA molecule base that coded for the amino acid histidine. There is histidine over here. So we have the name of the amino acid that we are guided to focus on. And in this amino acid, we do have the anticodons. Now they are asking us for the DNA triplets. I'm happy to take 3 options for this one. So I'm going to take option A, option B, and option C. Anyone, please give me your bases. I wish I'll chill. I will wait for you guys. I am not giving out answers today.
Okay. CAT.
Okay.
Okay, the first option, CAT. Did I get it right?
Yes, it is.
CAT. All right, thank you. Let me get a second option. Please unmute and tell me your second option, unless all of us got CAT. And I doubt all of us got CAT. Did we all get CAT, guys? Yes? Yes? Okay.
I got CAT, ma'am.
You got CAT also. Okay, let's figure it out. So they've given us the anticodons for histidine, and now they want DNA triplets. Now, before we get to DNA triplets, we need to be able to find messenger RNA codons first. So let's look at the sequence of the strands that are formed. Where does it all start? Let's remind ourselves. We start with DNA, and then we use our DNA to form messenger RNA. We then use codons from messenger RNA to form transfer RNA. And then from transfer RNA, we should be able to get our amino acids. From amino acids, we are going to bond those amino acids together, or they will be bonded together to form a protein. Now let's look at what we are given and what they want from us. I'm going to change my color. So what we are given here is an amino acid and a transfer RNA anticodon. What are they asking for? They are asking for DNA. So that means we first need to find the messenger RNA, which is here. That's what we need to find first before we get to our DNA. I said we're going to work a little bit. So let's find the messenger RNA codons that they didn't include there. So G will be complemented by C, and U will be complemented by A, and A will be complemented by U. Stop me if I'm wrong. Now, those are the codons of a messenger RNA strand. That's not our answer. So we have now found the codons of our messenger RNA strand. We now need to use those codons to pair them with bases that were initially in the DNA strand. So if this is CAU, that means our triplets will need to complement the CAU, meaning the DNA triplet C will be complemented by G, A will be complemented by T, and U will be complemented by A. Stop me if I'm wrong. This is your answer. GTA. I need to hear those voices. Unmute, let's speak. Unmute, voice, reaction, a hey, a hi, a or no. I need to hear something. Why are you guys angry at me? Are you not talking to me anymore? Let's not fight, please. I need to hear voices. I need to hear voices, guys. Let's talk.
Okay, ma'am. It was quite tricky, but we got it.
We got it.
Yo,
Yeah.
coming,
Yes, I want to hear those laughs. I want to hear everything. I want to hear everything, guys. Please. So this is what I assume you guys did. You said, oh, okay. They gave us the anticodons here for histidine, which is GUA, and they didn't give us anything here. So we are asked to look for something that is here, the basis there. But remember, these are bases for messenger RNA. You have to take it back to DNA. That's what they asked. So I'm happy that people are making these mistakes now, so that during your exam, you're going to read those questions carefully to make sure that you answer according to the question. We are going to do well in those May exams. I know we are going to do well. Okay, I'm interested to hear the answers for this one. I know we have not covered mutation, but I'm interested to hear. This is a bit tricky. So let's listen to maybe the answer. During the synthesis of the same protein, this will be unfair for me to really go hard on you guys because we haven't covered this together. But during the synthesis of the same protein, there was a change in the amino acid sequence because alanine was replaced by tryptophan. Explain the possible reason for this. Anyone who attempted this? They can just give us an answer, then we take it from there. Anyone who attempted this 3 months? Anyone at all? You guys are too quiet on me today. What's happening? Lulu, I'm listening.
Maybe this caused a change in the base sequence.
Okay, let's see. What did you say? This caused?
A change in the base sequence.
Okay, tell me what's the marker location for this question?
It's 3. 3? Yeah, 3.
3 marks. So judging based on your answer, how many marks do you think you'd get?
1.
1 mark. And remember, we have to fight for that 3 marks. We look at the mark allocation in order for our answer to be guided by, right? So the mark allocation says 3. So that means I need to give a little bit of juice for me to get the 3 marks. Now, this is mutation, and I'm going to hopefully find time to explain mutation in this particular lesson. So mutation is a change that, may happen during DNA replication or during transcription. Now, this is where the change happens. Remember, in our DNA, we find the bases. Please just mute for me a little bit while I'm explaining this. So for example, there could be an addition of a base or a substitution of a base. What do I mean by this? So if in a base, for example, in one strand, these are our bases. Let's say there is a T, there is an A, there is a C, there is a T there. Now, the transcription is going to use this template strand in order to form messenger RNA. And then the codons of messenger RNA will be used in order to form transfer RNA, which will be used to form amino acid and then form a protein. This order is going to be followed. Now, if there is a situation whereby there's a certain mistake or a change that is regarded as mutation. Let's say, for example, we were following the triplet TAC. Now, if TAC is our base for DNA, that means for messenger RNA, we will then have AUG. For transfer RNA, we will then have UAC, and then a particular amino acid, and then a particular protein that will be formed by the chain of amino acids. But now, if there is an addition of a base, let's say there was a mistake or a change whereby a base got added here, and that base is T, that will change everything. It will change the codons of the messenger RNA, which will change the anticodons of transfer RNA, and possibly change the amino acid that is formed. Where did the change initially occur? In the DNA, and it affected everything else because the codons and anticodons and amino acid are all determined by the code in DNA. So what are they asking here? They are saying during the synthesis of the same protein, during the process of making the same protein that you have on the diagram, there was a change in the amino acid sequence. Where's the amino acid sequence? There it is. This sequence of amino acids, meaning the order of which the amino acid are bonded for with each other, has changed. Then they become specific about which amino acid. They are saying alanine was replaced by tryptophan. Let's look for alanine here. So alanine is this particular amino acid. So they're saying this one was replaced by the same one here, which is tryptophan. So instead of having the amino acid alanine, histidine, tryptophan, leucine, lysine, we then end up having tryptophan, histidine, tryptophan, leucine, lysine. So where did all of this change? That's what they are asking you. Explain the possible reason for this. Explain the possible reason of why alanine would be replaced by another amino acid. How are we going to answer? There was a change in the bases of DNA. That was the 1 mark that Lulu got. But now if the bases in DNA change, it will result in a change in messenger RNA codons. That's your answer. A change in DNA bases or code will result in a change in messenger RNA codons, which will result in a change in transfer RNA anticodons, resulting in a different amino acid formed. So because there was a certain mistake or a change in bases that were coding for this particular messenger RNA, it led to alanine being replaced by this. I will cover it if we do have time at the end of our lesson and show the notes. I don't want to lie, this can be a bit confusing, but I think the example is not too bad. But yeah, I will cover it if we do have time. Okay, describe transcription. I think this one is a copy and paste and we have described it already. Let's do this one. Oh. Okay. Identify molecule X. We're back. Olile?
So, sorry, are we describing transcription?
We here. Are you able to see my screen 2.1? I skipped transcription because I said we've already described it in the previous question.
Oh,
Okay. If you want to mention your answer to check it, I'm listening. I'm happy to listen.
it is the process where mRNA is formed from DNA.
Okay, let me go back here. I can't really see who's talking. Please talk again so that I can see on my screen.
I'm saying I'm describing a transcription. It is the
Okay.
process where mRNA is formed from DNA.
All right, let's talk. What's the marker location for that? If you can see on my screen. For the question, they said describe transcription. What's the marker location? Are you able to see my screen?
It's 6, yes, it's 6.
6 marks. So from your answer, it is a process whereby messenger RNA is formed from DNA. I'm not sure if I heard you correctly. Or DNA, yes, messenger RNA is formed from DNA. Is that what you said?
Yes.
Okay, I want us to think about it. That is a one sentence. And the marker location for this question is 6 marks. So how many marks do you think you'd get from that answer?
I did not end there.
Oh, okay, I'm listening.
Yes. I said it takes place in the nucleus. The DNA, it takes place in the nucleus, which is the transcription. The DNA double helix unwinds and unzips. One DNA strand acts as a template. Complementary RNA nucleotides pair with exposed DNA bases. Then mRNA is formed.
Okay, let's go through it together so that I can tell you the marks. Let's read again. The first 2 points, no marks for it, because they want you to describe the process itself, not where it takes place. So nothing about the location and all of that. So a process whereby messenger RNA is formed from DNA, that's a definition. They're not asking us to define. So that point will be incorrect. And the second point about it takes place in the nucleus will be incorrect. So we'll just probably cross that one out. But let me go to your third point. Please read your third point for me.
My third one, it says one DNA strand acts as a template.
Thereby the double helix.
Oh, the double helix unwinds and unzips.
Did you say double helix unwinds or DNA double helix unwinds?
No, the DNA double helix unwinds.
You get 1 mark so far. Then unzip, I would probably penalize you for unzip. I addressed this, I think, in the first question. The molecules bonds will need to break first before the molecule unzips.
Before the unzips, yes.
So I would penalize. So far, you have 1 mark. Yes, you can continue.
Okay. Then I said the weak hydro. What now?
I probably disturbed you. So after that, you said the DNA double helix unwinds and unzips. Then you mentioned something about 1 strand of DNA is used or something.
As a template, yes. Template.
Okay, then that's the second mark.
Then complementary RNA nucleotides pair with exposed DNA bases.
No mark there.
Then mRNA is formed. Then the last one, I said mRNA detaches and leaves the nucleus through nuclear pores to the ribosomes.
3 marks. Nothing more.
It ends there.
So I'll give you 3 marks. Now, this is the process where I said it's a copy and paste. And I think when I was teaching it, I did mention what you need to say in order to get the 6 marks. I did say that they will ask you to describe the process for 6 marks. I'm trying to go to that slide. Just in case it's blank on your side, just know I'm trying to allocate the slide so that we can speak about it. Unfortunately, life sciences is quite unfair. If you tend to rephrase and they feel like your sentences do not that they're not correct, but they're no longer explaining the process accurately, you will be penalized. I didn't hear anything about the RNA nucleotides from the nucleoplasm. That's what we want to hear. So I didn't hear that. That's why I'm giving you 3 marks only. But just give me a second. I'm trying to allocate that slide so that we can speak about it. We need to learn these processes. They are going to ask you to describe DNA replication or transcription or translation. I'm not sure if you can see my slide. I have transcription exam guidelines. I will need a few people to thumbs up if they can see that. I've left the question paper for now. So if you can see the DNA, no, not DNA. Okay, cool. A few thumbs up. Okay, let's look at this. I spoke about it. Okay, so you need to say the double helix DNA unwinds. Sorry, let me get my pen. And I said here, if you just mention double-stranded, you won't get a mark. It has to be double helix DNA unwinds. And then even on this slide, I think it's the other slide that is correct. Yes, there's a slash here, but please, you have to speak about the bonds first and then the molecule unzips. Then one strand, the word one needs to be there to act as a template. This one is correct, to form messenger RNA using free RNA nucleotides from the nucleoplasm. I didn't hear this part. Okay, then mRNA is complementary to the DNA. I think I did hear that. Anything about the RNA moving out of the nucleus, you won't get a mark for it. We don't allocate marks for this one. Now, you can be lucky where they allocate a mark for unwind, for the hydrogen bonds, and unzipping, then end template to form messenger RNA strand, and then this. With just these 5 points, you would have gotten yourself 6 marks already. So anything else that you say before or after that is not correct, you won't get a mark for. So the best is to try and remember this and make that change. Please start with the hydrogen bonds and then the unzip, because you don't know how they're going to mark it in June. They can allocate 2 marks for it or 1 mark. I also don't know. We only find out on the day. I hope that makes sense. I think that was Olile. I hope that makes sense. Olile?
Yes, thank you. It makes sense.
Okay, thank you so much. Thank you for checking your answer. All right, let's
Thank
try
you.
and
Sorry,
push,
ma'am.
guys. Yes, yes.
Sorry, ma'am. There's a point. Is it important? There at the last one, it says the mRNA moves from the nucleus to the cytoplasm.
There's no mark for
It
that.
attaches to the yes. There's a mark for it, or it's just there.
No mark for it.
Okay.
No mark
All
for
right.
it.
Thank you.
Thank you. Okay. If you have to sing these points or whatever, I don't know. Just please remember them. 6 marks. We need that 6 marks. We have to fight for that 6 marks in the exam. Please, please, please. I'm trying to go back to the questions. Please let me know if you can see the questions. Just a few thumbs up. Well, let me know if you can see the questions. I'm back to the questions now. All right. Thank you, Nantanka. Thank you, guys. Okay, let's do it quickly. That's another protein synthesis. We're not going to panic when we see protein synthesis question. We're going to answer these questions head on because we know our content. Identify X and Z. Molecule X and Z. Okay, I'm not happy with the word molecule, but here. Anyone, please unmute and speak. Let's move. Let's move. Let's move. Or should I call? I'm listening. Olile, I'm listening.
X is amino acid
X is amino acid. That is correct.
and Z is messenger RNA.
Z is messenger RNA. That is correct. Thank you. Then give the nitrogenous base sequence of the DNA base triplet complementary to the middle codon of molecule Z. Ooh, what do they want here? For 2 marks. Are we working or not? Give the nitrogenous base sequence of the DNA-based triplet complementary to the middle codon of molecule Z. So middle codon, let's figure out the codons. So this would be the first codon, this would be the second codon, and this would be the third codon. They want us to focus on this one because they said it's the middle one. They guided us, guys. They said molecule Z. This is molecule Z, and this is where we find the codons. So anyone to give me the bases there? Let's see. Wavudi. Let's speak, guys. I don't want the same people to be giving me answers. There's also other people that have not spoken. Anota, I haven't heard your voice in this lesson. I need you to answer also. Okay, let's answer, guys. Ntokozo, I haven't heard your voice. Please. Let's answer. Let's answer.
Okay, ma'am, I got ATC.
You got A, T, C. DNA molecule Z. So A. You got ATC.
Yes.
Okay, I'll take that as my first option. A, T, C. Anyone with a different triplet? Ooh, sorry.
Okay. Okay. What I
I'm
got
listening.
is TAC.
T, A, C. Anyone else with a different triplet? Let's figure this out. What are we given? A messenger RNA. What do they want? DNA-based triplets. So we are just going to use these bases, and they said the middle one. This is the middle one because this is codon 1, the first one, the second one, and the third one. This is the middle one. So we are going to pair these bases. We are going back to DNA. So if we have to have, if we were to have our sequence, sorry, we will start from
Sorry,
having
ma'am.
DNA. Yes, I'm listening.
Okay, sorry, it's ATG.
ATG. Okay, noted. So let's see what we have here. So in terms of the sequence of molecules that are formed here, we use DNA to form messenger RNA. Oh, my pen is not writing. Okay, we use DNA to form messenger RNA. We are given messenger RNA, we need to go back to DNA. So we are going to pair these bases. A will be paired by T. We are moving back to DNA. U will be complemented by A. And then G will be complemented by C. Our answer is TAC. How did you get ATG? I didn't see who answered ATG. Please explain to me how you got ATG so that at least I can see where the mistake is.
Okay, ma'am. I think maybe I did transfer DNA.
You did transfer RNA.
RNA, yes.
Okay. All right.
Okay.
So let's take note of the words that they use in the question. They use the word codon, and they said the middle one, and they said molecule Z. So they've told us which bases they want, which is the DNA base. Okay, so we just need to keep on practicing these activities. We will be fine. Then bases at Y. The bases here, Anota, please give me the bases.
C, U, G.
Bases at Y. Okay, the first option.
Oh, sorry. Y, sorry. At Y, it's going to be GT A.
GTA. That's our first option, guys. Thank you, Anota. Anyone with a different base triplet? Anyone with a different sequence? So far we are having GTA. We are answering 212B. We are looking for the bases at Y here. So the first option we have is GTA. Anyone with a different sequence? Please unmute and tell me. Did you guys all get GTA?
I think, sorry, I wanted to correct my mistake. I think that it's supposed to be U instead of T. I mean, not, yeah, U instead of T, sorry.
So you're correcting, you're saying it's not GTA, it's GUA.
Yes.
Correct, it is GUA. C will be paired with G. A will be paired with U. U will be paired with A. Now I'm worried because we said it was GTA, our option, and I asked, is there anyone with a different sequence? And then there was nothing. So that means people, by being silent, they were telling me that they got GTA also. Am I right to analyze it like that, guys? Or we were just not answering? Maybe because we're tired or anything of that matter, or we're questioning our basis. I want us to get this right. So what are we having here? We are having a messenger RNA strand. What is this? These are anticodons in a transfer RNA strand. There is only one molecule with this shape. It's a transfer RNA. So we are pairing a messenger RNA with a transfer RNA, hence the U. So it can't be T there, where they didn't ask us about DNA. We answered the DNA question in A. So here at Y is the anticodons for transfer RNA. I need us to get this, guys. I need us to get this. It's not that difficult. We have to be able to figure it out at all times and do these activities. Because also, if you didn't do the activity, you'll probably be confused now. Okay. The teacher is very sad. Name and describe the process shown in the diagram during the formation of protein. I need someone to assist us. Let's look at the mark allocation. 7 marks. We're not skipping this one. We need that 7 marks. We need that 7 marks. Tandazo, what did you say? I'm listening. Okay, anyone? Guys, are you still there or am I just talking?
Can I try, please?
Yes, please.
Okay. The name of the process is translation.
Check.
Okay, number 1. Each transfer RNA, each tRNA carries a specific amino acid. Then the anticodons matches with anticodons of tRNA matches with codons of mRNA, and I'm not sure if maybe I'm saying it correct. Then another point, tRNA brings the required amino acid to ribosomes. Then amino acid bonded attached by peptide bonds to form required proteins.
7 marks. Again, this is a copy and paste. Each transfer RNA carries a specific amino acid, 1 mark. Why is it 7 marks and you are seeing 6 ticks on the board? 1 mark was for naming the process because the question said name and describe. So you first have to name and say translation, you get a mark. Then, sorry, the other 6 marks is for describing the process. Now, these are the points for describing translation, nothing else. The transfer RNA carries a specific amino acid this side. But this is when the anticodon at the bottom of the transfer RNA matches with the codon from the messenger RNA. Then the transfer RNA will say, oh, okay, cool. I know exactly which amino acid to bring. So the transfer RNA will then bring that amino acid to the ribosome. It was told by the code from the messenger RNA, this is the code. This is what you need to bring. It goes and collects, and then it brings it. So it will be one amino acid. It will do the same thing for the second one, for the third one, up until we form a chain of minimum 50 amino acids that are bonded to each other by peptide bonds. This will be the amino acid brought to the ribosome, and another one, and another one, and another one. These lines represent the peptide bonds that are attaching the amino acids to each other. That's how we form the protein in translation. 7 marks. Questions on that? I'm happy to take questions. DBE, June 2023. This was the question. The table below shows the codons, that code for some amino acids. A mutation, remember that change that I spoke about, that I said I haven't covered yet. The mutation caused a DNA-based triplet to change from CTG to CTA. Describe the effect of this mutation on the protein that is formed. I want us to work together to figure this out. Please, if you can make notes on your side, as I mention or explain this answer, and please look at the marker location, 4 marks. Now, I said to you, a mutation happens in the DNA. And the mutation is a change that occurs in the bases of the DNA. So if we follow our normal sequence, would be a change in DNA would result in a change in messenger RNA codons that are formed, which will result in the transfer RNA change of anticodons, which may result in a change in the amino acid that is formed. Now, in the amino acid may remain the same or may be different. It depends. And we are going to be guided by what we are given in the question. Now, depending on the changing of the amino acid or not the changing of the amino acid, that will then influence or determine the protein that will be formed. Please do write this down. I'm going to change my pen color. Let's follow what we are given. So what are we given here? They are saying a mutation caused a DNA-based triplet to change from CTG to CTA. Now let's follow this together. We are going to first find the name of the amino acid that is formed when the bases were CTG. So let's go down here and write it. Let's figure it out. So for DNA, what is the base that we're working with? We're working with CTG. We are going to use that base to find the messenger RNA codons. So C in a messenger RNA will be paired with a G, T will be paired with an A, and G will be paired with a C. Stop me any second if you feel like I'm messing this up. I've been teaching from 6 a.m., so I could be tired, but I think I am paying attention, so these bases are correct. I'm saying so far, we are working on these bases before there was a mistake, before there was a change. Now, it's up to us if we skip transfer RNA or not for this question. Why am I saying that? we need to find the name of the amino acid that was formed. And in our table, that is guided by the messenger RNA and not necessarily the transfer RNA. So we can use these bases of the codon and messenger RNA to find the name of the amino acid because that's what we are given in our table. So GAC, let's look for GAC. GAC, where is it? There is GAC there. What is the name of the amino acid that is formed? Aspartic acid. We write it down. Like I said, we can skip transfer RNA or we can even write the base of transfer RNA. I actually don't care. Let's write them. CUG. What is the amino acid that we got? It is that aspartic acid. So A, S, it's going to be a bit slow. P, A, R, T, I, C, acid. Okay, now obviously we don't know the name of the protein that is formed, and it doesn't really matter in this case. It's not going to change anything about our answer. We go back to the question. They're saying a mutation caused a DNA-based triplet to change from CTG to CTA. This is the amino acid that was formed when our bases were CTG. Now let's find out if the amino acid would change or remain the same since there was a change that occurred in the bases. Let's find out together. What was the change that occurred here? The bases then became CTA. So I'm sure you can see what happened here. The first two bases didn't change. C and T are still there. What happened here? G was replaced by A. That is the change that occurred. So how does that affect the messenger RNA codons? If we're having CTA, the messenger RNA codons are going to be GAU. Now, once we have the messenger RNA codons, we are able to go to the table and find out the name of the amino acid that is formed. Let's look for GAU in the table. There is GAU over here. What is the name of the amino acid that is formed? Aspartic acid. We write it down here. Aspartic. It's not going to write the whole thing. Acid. Now, I need us to unmute so that we can be active on this one. When we had CTG as our base, aspartic acid is the amino acid that was formed. Then a mutation happened whereby a G got replaced with an A. The amino acid that got formed due to this change was still aspartic acid. Do you think the protein that will be formed here, will it be a new protein or the same protein that will be formed? Give me an answer. Will it be a new one or the same one? Based on what I've explained. Anyone?
It'll be the same protein.
Okay, I'm taking that. It will be the same. Why are you saying it will be the same?
Isn't it because we're still having the same amino acid?
Thank you so much. Same protein. Why are we saying the same protein will be formed? Because we are still having the same amino acid formed. There was a change, but we still form the same amino acid because it is possible for an amino acid to be coded for by more than one codon or anticodon. So we first had aspartic acid, there was a change, we still were able to form aspartic acid. So there was no change in the name of the amino acid. We still had the same amino acid. That means there won't be change in the protein. It will still be the same protein. But if we had an example whereby aspartic acid was the one that got formed, but due to the change, we ended up forming valine. So that means the amino acid name changed. From aspartic acid, we then formed valine because of the change. That will lead to the protein being different. If the amino acid names remain the same, even after the change, the protein becomes the same. If the amino acid names change due to the mutation, the protein will be different that will be formed. Now, how do you answer to get 4 marks? Exactly what I've written down, but you write it in sentences. Mutation occurred in DNA bases whereby CTG was replaced by CTA, 1 mark, which meant that messenger RNA codons changed from GAC to GAU, 1 mark, which led to the amino acid changing, or the amino acid aspartic acid remaining the same because these two bases are coding for the same amino acid, 1 mark, which resulted in the same protein being formed. That's another mark. That's how you get 4 marks. Can I get a response of some sort, guys? Just a a hi, a yes, ma'am, no, ma'am, something. Something, please, 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 please. Okay, I'm getting reactions. I'll take that reaction. I'll take it.
Yeah, thank you, ma'am. You are keeping us on our feet.
You guys are not responding to anything, so I'm not on my feet. I'm actually under the table because I'm getting stressed out. There's no
We are on
responses,
our
there's
feet.
nothing.
We are on our feet.
You are on your feet. I'm under the table.
You are under the table. Okay, you can
I
come
am
out.
under. Literally, guys. That's why I'm saying sometimes just unmute and say yes, ma'am, or something. Because I feel like I'm just talking and talking. And maybe you guys are not getting these things. Okay. Thank you for that. All right. I'm no longer under the table now. I'm back up.
Okay.
Okay. Let me drink my water. This is protein synthesis. I want us to get that full 8 marks. Look at the question again. If you have to go over your answers, please do. I'm having a 2 minutes break to get more water. So we are doing 1.5. I want all of us to get this 8 marks. This is from June 2023. You are writing a June paper. So this could be a diagram that you get in your exam. So I'm having a little break, 2 minutes. Look at the questions, look at your answers again. We've gone through these questions now. You should be seeing where you've made mistakes and then you rectify your mistakes. So 3 minutes, well, yeah, it went from 2 to 3 minutes, but yeah, 3 minutes, I'm gonna get water and then we start marking. Okay, I'm back. All right, let's do this. 8 marks in 2 minutes. Let's do it very quickly. Janita, let's do this. Let's do this. Identify process Z. Let's move very quickly, guys. Janita, this one is for you. Am I back?
Yeah,
Yes,
ma'am, I'm stuck.
Where are you stuck? Where are you stuck? Okay. Let's get someone else who can help us. Anyone? Anyone?
Ma'am, it's transcription.
I think that's Anota. Okay. Let's look at this. So Anota, you're saying process Z. Let's start with the first one. Process Z. There is process Z. You are saying that is?
Transcription.
Transcription is correct.
Molecule R.
Molecule R. Yes, I'm listening.
mRNA.
mRNA. Let me break this down before I continue. This diagram represents protein synthesis. And you should be able to see that both processes or stages or phases of protein synthesis are represented here. This first part is representing transcription. And you are able to see that by these lines. That's a nuclear membrane. So this is taking place in the nucleus. And then they're showing you an arrow here to say, oh, okay, this molecule R that is formed here, see all of these dashes, they are showing you that it moves out of the nucleus through the nuclear pores, and there it is, it's attached here. So you are able to see what is happening in the nucleus and what is happening in the cytoplasm. So both transcription and translation are being shown here. Organelle Q. What did you guys say? There is organelle Q. The organelle in the cytoplasm where messenger RNA attaches itself. Anyone, guys? Let's move, please. Just unmute and tell me. Lulu?
Ribosome.
Ribosome. Ribosome. Give the collective name of nitrogenous base O. There is O here. So they want the collective name of this. Is it codon or anticodon? Anticodon. Thank you. Anticodon. Let me go back to Janita. Are you catching up? Because you said you were a little bit confused. How is it now? Is it getting better? Or are you following?
Yes, ma'am, I'm following.
Okay. Thank you so much.
Okay.
Yes, I'm listening. Do you have a question?
Oh, you said it's an anticodon. Is it on, because it's on a transfer RNA?
Thank you so much.
Okay.
It's going to transfer RNA. Determine the sequence of nitrogenous bases at area S. Ooh, where is S? There is S over here. Nitrogenous bases, I am getting AGT. AGT. Did you guys also get AGT? AGT. Unless I'm wrong. I am right. Okay, I'm not getting any reaction, guys. Just unmute yes or no. I'm getting AGT because I'm reading it from top to bottom. I'm not sure if you guys read it differently. I'm seeing there's a message, but I couldn't read it. I'm reading it. Okay, thumbs up. Okay, I've read it from top to bottom, hence the bases that I got. Okay, think you guys are happy with that. Which strand 1 or 2 was used as a template for the formation of molecule R? There's the strands, strand 1 and 2. Anyone unmute and tell me, which strand did we use in order for us to form molecule R?
1.
The double helix.
The double helix. From the double helix, there's 2 strands. There's
Oh.
strand 1 and strand 2. You need to tell us which one was used between 1 and 2. I think I heard an answer. Someone said 1. 1 is
2,
correct.
ma'am.
2. I'm hearing a 2. The
Oh, no, my
strand that
mistake,
was
my
used,
mistake.
guys, is this one. It's strand 1. This was the one that was used. How do we figure this out? So remember, they're asking us which strand was used to form molecule R. We can either use this part as a guideline or this part as a guideline. So if we are using the bases here, A, U, C, we need to check which bases from these strands would be complemented by A, U, C. Let's look at strand 2. Will A be complemented by A or will A pair with A? No. So already strand 2 is out. But if we look at strand 1, T will be paired with A, that is correct, or complemented by A. A will be complemented by U. G will be complemented by C. So we use strand 1. That's how we figure it out. Which amino acid, 3, 4, or 5, will be brought to area P? Which amino acid? 3, there is 3, there is 4, there is 5. Which one will be brought to area P? What did you guys say? I'm listening. This one you kind of have to figure it out. It's not going to be easy. It's not going to be, well,
AGT.
AGT. Which amino acid? So there's 2 ways of figuring this out, guys. Yes, I'm listening.
I can't remember on S you gave us
It's number
AGT.
4.
And it's on a strand 1. So that's how I got my answer.
Okay, let's just double check if we are answering the same question. 145 we've answered. We are answering 155 now. Okay, which amino acid from 3, 4, and 5 will be brought at area P? So between this amino acid and this amino acid 4 and this amino acid 5 will be brought here at P. Now there's 2 ways to figure this out.
It's number 4.
Okay. Will it be 4?
Yes.
Okay, let's see. How did you get 4? Tell us.
Because if you look at 5, 5 will pair with the middle one, and 3 will pair with the first one, and then 4 will pair with P.
Yeah, this is the easiest way to do it. 4 is correct. Now, the difficult way to figure it out would be to go back here, but that would be too difficult. So you'd come back here and then find the basis for S, and I think we did determine basis for S, AGT, and then use AGT in order to find the codon for R over here, and then from that codon, we match it with anticodon. Oh, that's a long process, and I think this one was better. So if you're looking for anticodons to complement here, that will be UAC done. Then 5 will complement GGU. Then 4, that's the only remaining amino acid, I guess. Okay, name the type of sugar that forms part of the structure of molecule R, ribose sugar. Okay. The teacher's energy is at 0. Describe the process of DNA replication. How many marks? 6 marks. 6 marks. That's a lot of marks. And those are marks that we need, guys. Those are marks that we really need to get. I'm listening. Nom Dani, so I need to hear your 6 points. for 6 marks. Nomdaniso. Okay, I can hear that's Tanda's, all right?
Yes, ma'am. My first point is DNA double helix unwinds. And then it says weak hydrogen bond break into two separate strands. Both strands act as a template. Free floating DNA nucleotide from the nucleoplasm joined to their complementary DNA base triplet. Hydrogen bond reform, DNA rewinds and rezips to form two identical DNA molecules.
6 marks. 6 marks. Okay. For the rest of the activities, I'm going to send a memo. Unless there's a specific activity from the ones that we haven't answered that you would like me to do. I can try and do one more. You can tell me which one you found a bit difficult. Then that's the one that I can do. I can only do one more. Or I can just send you guys the memo for all the other activities. What do you prefer? Should we do this last one? I'm not sure if you can see this one. Last year's one, June 2025. I'm not sure if you can see it on your screen. Then we
Yes,
call it
please.
nights. Yes? I didn't hear?
Yes, can you please do it?
Okay. Let's do it quickly. The diagram below represents a stage of protein synthesis. Each amino acid PQRS is coded for by a different codon to the messenger RNA. Identify the stage of protein synthesis represented here. Translation is your answer. Because that's easy. We're able to see transfer RNA and messenger RNA and the ribosome. Translation. Identify molecule 1. Now I need to figure out where is molecule 1. Okay, there it is. Molecule 1 is transfer RNA. Organelle Z, there is organelle Z. That is your ribosome. State the sequence of nitrogenous bases on the DNA molecule for codon 3. Codon 3 DNA bases will be C, G, T. CGT. For 4, oh, now I have to figure it out. 2 marks. Anyone that got 4 while I'm figuring this out? Because we'll have to use the amino acids here. We'll have to use the transfer RNA, then find messenger RNA, and then find DNA. Okay, let me do it quickly. So the amino acid for UAC to U will be A. Where is AUG? So this amino acid will come here. UAC, AUG, okay? Then for GCA, CGU, this one will come here. Then CGA, I need the one that starts with a G, GCU, this one will come here. So the one that we have not used is R. We haven't used R. And with R, we are getting, yes, R. Okay, so the amino acid that we have not used here is R, which has transfer RNA. Okay, wait, my pen. So what we are getting here is transfer RNA. We will need information about messenger RNA and then DNA. Okay, so the transfer RNA that we're given from amino acid R. Remember, we started by eliminating the obvious amino acids. They want us to get the DNA base triplets, but we first need to get which amino acid has the correct anticodon for us. And this is amino acid R. So that means the anticodon there, which is given, is CUA. So now that we have transfer RNA, we are on our way to getting the correct answer because we just get messenger RNA and then DNA. So I've done it for you guys. So CUA, the messenger RNA codon there will be GAU, meaning the DNA base triplets will be CGT. CTA. CTA is your answer. Wow, we had to work a little bit here. Last one. Which amino acid PQRS is coded
Can I just
for
ask
by
a question, ma'am?
Yes.
For B, do you need to show your working? Do you need to show how you got to the answer, or can you just write the answer?
Just write the answer.
Okay.
Is it CTA or GTA?
CTA. CTA. What did I want to say? Oh, the last question. Which amino acid PQRS is coded for by codon 5? I think we kind of have answered that. Which amino acid P? There's the amino acid. There's my arrow. Protein synthesis. Okay. All right. That's it on my side. There's no time to do mutation. So in the next lesson, we are going to do mutation. It will probably take me 5 to 10 minutes to just cover mutation from the notes. And then we do our new topic, meiosis. So this is the order of the topics that I'm looking at. Time is a problem, but I guess we just have to work with what we have. So we're going to do meiosis, and then straight after meiosis, we do genetics. That's a big topic. So it's going to take us a little bit of time to get through it. And then we'll decide from there, maybe human reproduction, because that's also big. I do not see us covering evolution at all. I see us doing questions for evolution, but there it's a read. You read. That 54 marks, you kind of need to read and figure out what is happening. If you have to watch videos online, you might need to watch videos online because we won't have time for it. At school, we cover it over 4 weeks, the whole month. That's 4 hours per week. So that's 4, 8, 12, 16 hours. We're not going to be able to do that in the lesson. So please do. I think I did send the study guide for evolution. If I didn't, please just remind me on the group and I will send it. But please start studying evolution and endocrine system, all the other topics. If you are still not spending a minimum of 2 hours in life sciences a day, you're not going to be ready for exams in May. And I know sometimes it's not possible to do it during the week, but then find time over the weekend at least to catch up. That is very, very, very important. But yeah, I'm happy to take any question.